My name is Gabriel Antonio Minero, and welcome everybody to my talk about integrated amplification and detection of pathogen nucleic acids using magnetic biosensor. In my talk, I will concentrate on detection of tuberculosis pathogen infections. And to begin with, I would like to show some numbers to prove that the incidence of tuberculosis is still a health concern in some countries, so to say, developing countries. And this can be a significant burden for population with secondary infections or health concerns such as HIV or diabetes. And the tuberculosis is a respiratory disease, just like coronavirus. It can be spread easily, due to migration or tourism flights. In 2014, they estimated a total of 330,000 tuberculosis cases reported by World Health Organization in the European regions. However, that represents only 3% of the global tuberculosis burden. And the reason for that is perhaps the absence of efficient diagnostic tools to analyze and foresee the disease. What they do in schools, for example, in certain countries like in Russia, where I am from, is a so-called tuberculin skin test reaction. And they inject tuberculin under the skin and they measure the size of the allergic reaction, skin reaction. Yeah, and then they, based on the history of a patient, they can prognose whether there is a risk or not for tuberculosis infection. However, that has been proven to be very unspecific and by far not sensitive. Tuberculosis can also be latent disease, which means it can be present in a human body without giving symptoms. And when symptoms come, tuberculosis can be suspected on the X-ray of lungs. And then a culture test should be made to confirm growth of Mycobacterium tuberculosis on agar plates. But that takes about 4 to 6 weeks to get colonies. Tuberculosis is extremely slow growing pathogen. And while the culture test may confirm the presence of Mycobacterium tuberculosis, additional screening for antibiotic susceptibility must be administered. But that takes another week, so the patient will be administered to take first-line rifampicin antibiotic. And I think that is extremely dangerous because if the strain of Mycobacterium tuberculosis is resistant to rifampicin, such treatment will only cause harm to the patient. The antibiotic resistance of tuberculosis is one of the complexity of tuberculosis-caused infections, and that mainly arises from mutation in certain genome. Another complexity is the formation of biofilms and presence of so-called persistent cells. And biofilms are the communities of bacteria covered in extracellular matrix so that they can keep nutrients close by the bacteria and be protected from toxic environment. That can be, for example, caused by inappropriate treatment using wrong antibiotic or wrong concentration of an antibiotic. but there are also other reasons for this kind of complexities. And the way to diagnose tuberculosis as well as to handle these complexities can be molecular diagnostics, where we look at the nucleic acid content of the patient samples, such as sputum or liquid biopsy. The nucleic acid content will be primarily double-stranded genomic DNA, whether it originates from bacteria itself, like from planktonic cells, or whether it's present in the extracellular environment, such as in biofilms. And using amplification techniques, for example, polymerase chain reaction, we can amplify this nucleic acid content, knowing the sequence, and we can design the right primers. But we will need to have right tools, for example, thermocycler for PCR. In order to separate strands, we need to anneal the reaction at 95 degrees and then control the temperature between 95, 55, and 72 degrees to perform the steps of PCR. Moreover, the DNA sample must be clean in order for the Taq polymerase being able to synthesize the bases. So even though this is a gold standard, already at this stage there are certain complexities and certain prerequisites for this technique to work. Furthermore, it's important to know what actually we investigate, whether it's genomic DNA that undergoes mutation, whether it is RNA, and whether this DNA or RNA comes from planktonic cells or from extracellular matrix. And for small molecules such as microRNA, polymerase chain reaction is not well suited because it requires additional steps to convert RNA into complementary DNA library and some other steps, which makes PCR less attractive for this target. Then another aspect while designing the biosensor is to think about signal readout strategy. And for the tuberculosis diagnostics, there is a strategy to detect binding event of amplified DNA with complementary probes immobilized on a paper stripe. Then there is the possibility to get multiple probes immobilized for multiplex analysis, and it's possible to detect the product by naked eye, which makes the method semi-quantitative. But the only disadvantage, perhaps, that this is quite expensive. It's $35 to $80 per test. And that even though the provider claims the result in approximately 2.5 hours, in the original paper, the binding event is recommended to take place for 5 hours to get better sensitivity. So it is time consuming and still laborious because all the steps such as isolation, amplification, binding, washing, and stuff must be performed by a real person. A slightly faster and less laborious technique, real-time PCR, actually gold standard technique in hospitals, is based on detection of fluorescence while amplification takes place, so that fluorescent dye intercalating into the double-stranded DNA lights up, and a certain amount of copies can be detected at certain threshold cycle of PCR, which makes this dose dependent But the disadvantage of this technique, main disadvantage, is a cost ranging for this PCR machine from $50,000 to $90,000. Like I said, it is commonly accepted and implemented in the center laboratories in the Western world, but it is way too expensive for developing countries. And there are alternatives to PCR. There you can find lots of publications about recombinase polymerase amplification, loop mediated amplification, and the rolling circle amplification. And all of them can operate at isothermal condition, means at a uniform temperature. They therefore do not need specialized equipment such as thermocycling, and can be applied in the lab on a chip systems, so they can be integrated. This talk I will concentrate on rolling circle amplification, but it's not to undermine the other techniques. Next in my talk is optomagnetic biosensor as an alternative for paper or fluorescence techniques. Um, let's speak about optomagnetic technique that has been developed at Technical University of Denmark by the PI Mikkel Hansen and a colleague of mine, Jeppe Fogh. Um, the technique is based on anisotropy of magnetic nanoparticles that have this ellipsoidal shape, and they are able to follow oscillating magnetic field. Depending on their hydrodynamic size, they will align along magnetic field with a different to a different extent, so to say. On the plot in the left corner, you can see the oscillating magnetic field as orange and the second harmonic optomagnetic effect as a blue curve. And it is second harmonic pretty much because it doesn't matter whether the field is at the positive or negative amplitude. The dipoles of magnetic nanoparticles will align to the field in both cases, just in the opposite direction, but from the optical point of view, it doesn't matter. In both scenarios, light will go through. Whereas at zero magnetic field, the particles are pretty much randomized, their orientation is not aligned, and they block light. The technique requires basically just a laser or LED, two coils to create a selective magnetic field, and the photodetector to detect voltage or light intensity passed through the suspension of magnetic nanoparticles. Then, through a Fourier transformation, we get the plots on the right side. I will speak in more details on the next slide how do we get them. But important is just to see the real part of the magnetic spectra for now. It's top plot. Here we compare 100 nanometer and 80 nanometer magnetic nanoparticles. They have a slightly different frequency at which the curves have a maximum value of light intensity, and that is because the smaller nanoparticles can follow, so to say, magnetic field of higher frequency than the larger nanoparticles. Okay, so here perhaps we can see it in a bit more details. Again, the plot of magnetic field is given as a sinusoidal line, and the optomagnetic spectrum I mean the light intensity passed through the suspension of magnetic nanoparticles, is given at the top a different frequency of the magnetic field oscillation, while at low frequency there is a the magnetic nanoparticles can perfectly follow the field, and there is a significant optomagnetic effect detectable. As the frequency is increasing, the effect is decreasing, with almost no optomagnetic effect detected at 710 Hz. The reason behind it is that magnetic nanoparticles keep a random orientation at the highly oscillating magnetic field. See, this is how they change their orientation in a high frequency oscillating magnetic field. They almost stay randomized. But at low frequency, there is plenty of time for the nanoparticles to align with the magnetic field. The light is therefore passed through the suspension and is detected in photodetector. So this is in a very simple words, explanation of the frequency dependence of the optomagnetic phenomena. Using trigonometry, it is possible to write the optomagnetic, to decouple optomagnetic effect as real and imaginary components of the light intensity. There's some math and Fourier transformations behind, but what at the end of the day we get spectra consisting of real part, imaginary part, and phase. And the phase is just mathematically calculated as as the arctangents from real part divided by the imaginary part. It's what MATLAB does. All of the three components of optomagnetic, or all of the three outcomes of the optomagnetic biosensors can be used to quantify signal. To what extent, I mean, to quantify hydrodynamic size of nanoparticles, or DNA concentration in response that cause changes in the hydrodynamic diameter. Here's the Brownian relaxation frequency that is very important as it is inversely proportional to the hydrodynamic diameter or hydrodynamic volume of the nanoparticles. And one more time, I give the credit to these two wonderful people, and personally I find this technique very powerful for characterizing magnetic nanoparticles and implementation of them in nucleic acid diagnostics. Let's move on from theory to the practice, and the following slides will speak mainly about our latest publication on automated on-chip analysis of tuberculosis drug resistance mutation with integrated DNA ligation and amplification. And here the keywords are the detection of mutation and the assay integration, so that less laborious work is required for diagnostics. This is our ultimate goal. Today, I'm speaking about implementation of optomagnetic technique with rolling circle amplification, but this is not to undermine the importance or effectiveness of other amplification techniques. It's just rolling circle amplification is particularly suitable for genotyping and detecting single mutations. So how it works? From the name of the technique, it's clear that it's about a circular DNA. but the circular DNA is formed in the very first ligation step. A special probe called padlock probe is designed to match sequence of interest in the target DNA molecule. And upon hybridization of the padlock probe, the proximity of 3 and 5 prime end can be used to ligate the circularized DNA molecule using enzyme ligase or amp-ligase. And then after joining the 5 and 3 prime ends and forming circle, which will only take place in case of perfect match on both sides of the padlock probe, another enzyme, DNA polymerase 529, is employed to synthesize the copy of padlock probe. But here it comes the secret or the advantage of 529 polymerase having a strong strain displacement activity. It displaces the previously synthesized copy, thus creating a concatenator or repeating units of DNA as a single product. So this clump of DNA contains a unit of repeating sequence complementary to the probe. And this is perfectly suitable for optomagnetic biosensor because magnetic nanoparticles binding to this repeating unit of DNA will form cluster, and cluster have a different hydrodynamic diameter, so will be distinct from single magnetic nanoparticle. This is our base for detecting target DNA, first using sequence-specific ligation to check whether there is a mutation or not, and then using the repeating units of amplified DNA to form clusters of magnetic nanoparticles. And I will present two strategies for implementation of rolling circle amplification and optomagnetic detection. This is the first strategy on disk plastic chip. Why disk? First, as you can see, it has multiple chambers, so it allows multiplexing. And second, using centrifugal force, basically we can spin down liquids and start a reaction by motorized action of the setup. But what's important is to keep ligation reaction in one chamber and cocktail for the amplification in another chamber to allow ligation to work without the presence of nanoparticles or polymerase. And then after padlock is ligated to activate spinning and bring the all ingredients into one cocktail, thus starting amplification reaction. As the output of the lab view and in course of amplification, we can see the optomagnetic spectra changing in time. And here you have real and imaginary parts analyzed. In the real part, you can see at high frequency signal is depleting, whereas it's building up at low frequency. Well, this is because the single magnetic nanoparticles depleted in order to build magnetic clusters of magnetic nanoparticles. Just like I said before, that's what makes rolling circle amplification particularly suitable for optomagnetic biosensor. Well, the same you can say happens in the imaginary part. Then, using MATLAB, we can analyze and quantify, analyze the phase lag of the magnetic nanoparticles that is otherwise zero at the beginning of reaction, then it's building up as clusters of magnetic nanoparticles rotate slow. there is a phase lag that can be used to uh, quantify progression of rolling circle amplification. On this plot of kinetics, the first 20 minutes of ligation takes place. Then the temperature is changed within about 10 minutes because it's a hot air based heating. It requires equilibration and takes some time. And then the signal is slowly building up so that you can see the padlock probe has a sequence has specifically ligated towards mutated target as we designed them. And then in course of time, the circle was amplified, led magnetic nanoparticles to bind and form clusters. And it didn't happen for the wild type, which is good. This means that our technique is sequence specific and can be used for genotyping. In this strategy, the steps of rolling circle amplification are performed in one chamber chip in the setup suitable for simultaneous measurement and analysis of 4 chips like that. The chips are still made from PMMA using plastic layers and pressure sensitive adhesive tape. Is what I forgot to say in the previous slide. Is a very simple procedure which doesn't require any special conditions, not like clean room or anything like that. Can be reproduced on site in any environment. However, what is significantly different from a previous strategy is that here we implemented contact heaters made of aluminum plates, which made the heating much faster. See, in the previous strategy, I mentioned that the temperature equilibration was about 10 minutes. It's because ligation takes place at 37 degrees and amplification takes place at 50 degrees. But using aluminum hot plates in this strategy, the change in temperature took perhaps 1 or maximum 2 minutes, which is much faster and much more precise, and can perhaps explain why the mutant, why the difference in mutant and wild type targets was improved. So from the sequence specificity point of view, the results looked a bit better. This strategy, however, required a sort of pre-annealing of a circular padlock probe to the target. Otherwise, the 529 polymerase is known to digest single-stranded DNA molecules. That's why in the first strategy they were kept in separate chambers, ligation and polymerase. But if you would like to put everything together in one chamber, which apparently can be done, it was important to protect the padlock probe from digestion by 529 polymerase. Thus, we pre-annealed padlock probe to the target. This step can be easily carried over to the sample processing step. As you have probably noticed, the target DNA is single-stranded. So what is usually done is that genomic tuberculosis DNA is heat shocked, so it's annealed at high temperature and then cooled fast to cold temperature, and then such annealing step can be performed in the presence of the padlock probes. No problem. Yeah. Else, from the optomagnetic point of view, the technique and the analysis was completely carried over in the strategy. Same principle. We have used the phase lag for quantifying the kinetics of the reaction. And so in the next slide, I will show the results and comparison of these two strategies. show you the results of the dose response curve, in which we measured synthetic targets of DNA at different concentrations in a 5% fetabouvin serum, just to mimic like the environment, like to measure the output in the sort of close conditions as it would be if we got the real samples. So at higher concentration of the target DNA, the phase lag was increasing, which means the clusters, bigger clusters or larger, no, more clusters or larger clusters were formed at higher target concentration. And the standard deviations and accuracy was higher in this case where the aluminum heaters were used instead of the hot air. Nonetheless, the limits of detection were quite close, 2 and 5 picomolar. The time used for the essays was 65 and 95 minutes, but the multiplexing capacity was higher. In the disk case, it was 16 reactions could be analyzed simultaneously. The specificity was similar, perhaps a little bit higher in this case. We think that an improved heater or heating system can be implemented in this disk microfluidic scenario just to improve the reproducibility of results. But as a feasibility test, both strategies look attractive to us to go with. And it's important to keep in mind that I'm talking about integrated assays in which we make mixtures of the reagents and the target DNA that is in this case synthetic, but in future it should be from the sample. And then we just let the setup and device to handle it by itself using LabVIEW procedures. Let me just give a quick comparison to a few other examples of rolling stock amplification readout strategies. So a classic approach to detect rolling stock amplification is based on fluorescence intensity. Another is based on paper stripe and immobilizing the DNA product on the paper, washing and detecting how much DNA is bound to the specific probe. Like I was speaking for PCR in the beginning. Both of them are quite attractive, but they're non-integrated, which means that people had to do steps, ligation, amplification, and detection by themselves. And the limit of detection, well, in this case, fluorescence-based detection was, say, around 10 picomolar. And in the case of Pawan Kumar et al., paper-based detection, they detected it in nanograms, but I converted it and it resulted in about 0.5 picomolar in 20 microliter samples that they used. So, all in all, our strategies, they, 2 and 5 picomolar are perfectly in between these values. So, where are we? We have developed the biosensor to detect mutation in tuberculosis DNA responsible for the antibiotic resistance, and this is all in integrated setup within about 1.5 hours. The limit of detection of 3 picomolar is about 2 billion cells in milliliter, is of course very high and is limited to the linear amplification rate of Fórum stock amplification. Can be slightly improved, but it will not compete with PCR with regarding the limit of detection. But this is way more controllable amplification technique. It's not easy to get the false positive signal, and the sensitivity can be enough for example, analysis of the colonies, so that the antibiotic susceptibility test can be performed much quicker, or for example for study of the biofilms, where lots of cells come together and there is lots of DNA to analyze. Well, in conclusion, I would like to say that the optomagnetic biosensors that I have presented is attractive because it can be portable, and the isothermal rolling circle amplification assay can be not only integrated with the optomagnetic biosensor, but can also be integrated on a plastic chip, which makes it disposable and low cost. Different variants of rolling circle amplification to overcome linear amplification rate are compared, and the detection limit of 3 picomolar is obtained in 5% filter bovine serum. I would like to thank my group at Technical University of Denmark and the financial support from Danish Free Research Fund, and please contact me if you have further questions. Thank you.